Good afternoon, everyone. Before we dive into this project, a little bit of background about peritoneal metastasis for anyone who is not familiar with this disease. Peritoneal metastasis is a type of cancer that usually originates from colon or ovary and spreads very quickly to the whole peritoneal cavity, since it's very rich in blood vessels where cancer cells can easily travel through. And modern treatments available including cytoreductive surgery, CRS, where bulk tumor nodules are resected and followed by hyperthermic intraperitoneal chemotherapy, HIPEC for short, where it infuses anti-cancer cocktail into the peritoneum. And the current standard of care for PM is called FOLFOX, which includes fulinic acid, 5-FU, and oxaliplatin, which can induce immunogenic cell death that you will see in a few slides later. However, the prognosis after CRS and HIPEC combined is still not very promising. The overall survival and disease-free survival are only 36 and 14%. That is because this type of cancer is often diagnosed in a very advanced stage. So what makes it so difficult to cure peritoneal metastasis? One of the factors is the highly regulated immunosuppressive tumor environment. And here is a picture representing a tumor with its microenvironment, where we can see the tumor is regulated by the various surrounding signals, such as angiogenesis or influences from cancer-associated fibroblasts or tumor-associated macrophages. And as mentioned earlier, here in the immune system in the microenvironment is highly suppressed, mostly by T regulatory cells and myeloid-derived suppressor cells, MDSCs, which leads to an environment that favors tumor growth and progression, which is also known as a cold tumor. This immunosuppressive environment allows tumor to evade from being completely eradicated by our immune system. Thus, the high recurrence rate of 20 to 50% of this disease. So in order to tackle this cold tumor issue, we used Resiquimod, also known as R848, which is a small molecule immune booster to reactivate the immune system against the tumor. But R848 is poorly soluble in water and goes into circulation and gets cleared by the human body very rapidly, which results in systemic side effects and insufficient drug exposure at where it was meant to be. So to improve the delivery, we encapsulate R848 in liposomes with the addition of DS-TAP lipid, which is a cationic lipid to improve its solubility as well as peritoneal retention. After doing so, we firstly did a pharmacokinetic study. The blue line here represents free R848, and the red line showed the liposomal formulation. And we found the DS-TAP liposome showed about 20-fold increased dose exposure in the peritoneal fluid. And at the same time, we also saw a similar difference in the intraperitoneum cells. In contrast, the R848 concentration in plasma with DS-TAP formulation was much lower compared to the free drug group, which is due to the rapid absorption of the free R848 I just mentioned. So together, this PK data indicated a stronger intraperitoneum retention achieved by the DS-TAP formulation. Later, we measured the level of interferon alpha secretion using ELISA. And in the peritoneum fluid, the DS-TAP R848 induced 3-fold concentration at 1 hour time point than the free R848. While on the right here showing the interferon alpha level in plasma, there was no significant difference systemically. And interferon alpha here is a marker for immune system activation. And studies have also shown that interferon alpha has the ability to turn the immunosuppressive cold tumor into a hot tumor as an immunoactive. Next, are somewhat in vivo results. And here is the peritoneal metastasis model established by IP injecting luciferase expressing colon cancer cells. And before treating with our immune boosting formulation, we dosed the mice with oxaliplatin to expose more tumor antigen release by inducing immunogenic cell death, as mentioned earlier. And 1 day later, we treated them with PBS-free RA4A or DS-TAP RA4A, and the mice were monitored by IBIS throughout the experiment. Here at the bottom right, the free RA4A in blue first showed the inhibition of tumor growth, but it bounced back at around day 4 and showed basically no difference than the PBS group at day 13. Compared to that, our DS-TAP formulation in red can effectively inhibit the tumor progression and showed significant difference at the experimental endpoint, displaying the improved efficacy. And with this tumor inhibition data, we further look into the immune composition change in the microenvironment, where it was highly immunosuppressive. So after the same treatment with oxaliplatin followed by the RA4A in free form or DS-TAP liposome, the IP fluid was collected and analyzed using flow cytometry. The left 3 figures showed the CD4 and CD8 population, and the one on the right is the quantified data. And CD8 positive cells have been known to have a major role in eliminating cancer cells, which was greatly increased by the DS-TAP formulation we developed, demonstrating the anti-tumor immunity is activated in the peritoneal cavity. The ratio in the free drug group was lower than the PBS group here, but it was not significantly different. So what this data is telling us is that the DS-TAP RA4A formulation was able to boost CD8 positive T cell infiltration into the tumor microenvironment, and therefore the increased ratio of CD8 over CD4 T cells in the oxaliplatin and DS-TAP RA4A group was consistent with our enhanced efficacy data. So to quickly summarize this work, we first showed the DS-TAP liposome are favored for enhanced retention in peritoneal cavity, allowing for longer drug exposure. We also demonstrated that the DS-TAP liposomes encapsulating RA4A can promote anti-tumor immunity, increase interferon alpha secretion only locally rather than systemically. And last but not least, the tumor growth inhibition and reactivated tumor microenvironment indicated that this formulation in combination with oxaliplatin is very effective, which can serve as a supporting treatment that works well with the existing colorectal metastasis therapies by turning the cold tumor into a hot tumor. And last, I would like to thank my supervisor, Dr. Lee, and everyone in our lab for always being there for me, and all the funding agencies for supporting this research. Thank you for your attention. I'm now happy to take any questions.
Great, thank you, Po-Han. So we actually do have some questions from the audience, both of them having to do with your lipid formulation. So we're gonna go in order. The first one is, did you try different lipids other than DS-TAP?
Yes. So I will pull up the slides. So we did compare liposomes with different lipid components with different charges, and we monitored the distribution and retention in the peritoneal cavity. So we tried different. So we have neutral liposome and negatively charged liposome in our DS-TAP formulation, which is positively charged. So compared to the neutral and negatively charged particles, the positively charged DS-TAP liposome showed the greatest retention in the peritoneum area up to 24 hours post-injection. So that's why we select the DS-TAP lipid moving forward with our RE4A delivery strategy.
Okay. So a very similar question, I suppose, is that was asked is any other, well, I suppose, did you use any other cationic lipids for the liposomal formulation R848?
We do another commonly used cationic lipid is called DOTAP, which is also positively charged and has double bond structure. Although I don't know the real mechanism, but literature suggested that DOTAP exhibits higher toxicity than DS-TAP towards some immune cells, such as macrophages or T cells. And in this project, immune cells in particular are the delivery target for this formulation. So we chose DS-TAP for our particles to be more, how should I put it, immune cells friendly. Yes.
Okay. And so we actually have a follow-up question from your first question, which is, so why do you think, or do you have any kind of intuition as to why the permanent cationic charge improves retention within the peritoneal cavity? Or is that basically just essentially what you just said, which is that the positive charge prefers the immune cells, or the immune cells prefer the positive charge, so to say?
So the improved retention is basically from the from the positively charged particle. So the peritoneum membrane is negatively charged. So we give the particle locally with the positively charged formulation. So the formulation can stay there for a longer period of time compared to other, say, the negatively charged particles, which was cleared from the abdominal cavity area like very quickly.
Okay, that makes sense. It's a great answer. And finally, I see that Leah has raised her hand. So go ahead, Leah.
Thanks, Wesley, and thank you for the presentation, Po-Han. I'm curious, could you speak to why you used oxaliplatin, or whatever, sorry, the drug that you used over other chemotherapeutic drugs?
Oh, sure. So here. So the reason why we used oxaliplatin is to induce immunogenic cell death. So again, our therapeutic strategy is firstly to induce immunogenic cell death in tumor cells by oxaliplatin to promote more tumor antigen release. And with more antigen, tumor antigen released, those antigen will be taken up by the antigen presenting cells, which has been activated by our by RE4A delivered by our formulation. And this antigen presenting cells will then prime the T cells to be cytotoxic against the cancer cells. So this by combining our DS-TAP RE4A with oxaliplatin can synergize with currently used standard chemo drug to induce anti-tumor immunity.
Great, thank you.